And now we have a speaker from Mila, Maksym Korablyov, and he would provide a different approach to building combinations. So I'm really excited to learn about this work.
All right, I hope everyone can see my screen by now. So I will be presenting a work which is done by Recover Coalition. So Recover is acronym, which is Repurposing Engine for COVID-19 Emergency Response. And we have many collaborators, in particular, we pronounce this University of Cambridge in Relation Therapeutics. And the full study is funded by Bill and Melinda Gates Foundation. But today I will be presenting results that were done at Mila. So as Wingong mentioned, I'm really lucky to speak after him because I can really complement the talk. So he already talked that drug repurposing is fast, and we do have repurposed examples of repurposed drugs for many diseases. And basically, in order to basically look at how many drugs in total we have, right, if you want to do it in practice, there is a library called Reframe, which has 12,000 molecules. And basically, these are all of the FDA-approved drugs and also compounds in clinical trials which are deemed to be safe. It's a physical library. It can be shipped and tested. And basically, this was already defined, right? So sometimes when we have two drugs, right, and when we're adding them together, we can say that if with two drugs, we have better inhibition effect compared to what we would expect by a linear model, then they would be called synergistic. And if they are worse than that, they would be called antagonistic. So this is one of the results that is really, really motivational, right? So this is actually stage 4 cancer. It's not COVID, but this is a clinical trial that came out pretty recently, which is called iPredict. And what was done here is they took standard of care, right? So there was a cohort of 100 patients, which was divided into two halves. And one of the halves received standard of care. And this is basically what are the drugs that currently prescribed for against stage 4 cancer patients. And another group was receiving basically drugs that were described by a huge panel of 35 physicians, which was called cancer drug combination panel. So they were basically looking at molecular targets. They were looking at sequences of the proteins that particular human has. And then they were basically trying to tailor individual treatments. So what we have in yellow, right, what we have in blue is the initial group, right, which received standard of care. And in yellow is this group of the patients who did receive predictions from physicians. And as we can see on the left, we have a tumor-free progression, right? So they live longer without tumor. And on the right, when all of the patients in the control group did not survive, right, in the group which received this hand-prescribed drug combinations, many survived. And this is really, really significant because if you think about cancer field in general and cancer drug combinations, there were about 10,000 clinical trials on drug combinations. And if you can complement that field, that's very important. So while I'm talking, actually, we are running one of the studies on real cells in cancer, but talk is more about COVID. So I would more concentrate on COVID. So hopefully after COVID ends, we can talk a bit more about cancer because it's definitely a long-term goal. So if we want to find a drug combination that is useful for a particular patient, right, or for a group of patients, right? So if you just take pairwise combinations, then we already have 72 million, right? And if you take the most optimistic screen, which is not even the BSL-3 lab, but if you take a large-scale screen of, let's say, several eco machines, so eco machines are the small machines that pipette drugs, and this can do 5,000 a week. And it already takes 300 years to just screen these drugs or pairwise combinations. So we really need some fast technology which utilizes machine learning, which could do it faster, because this way it is, if you do it by hand, it would be too slow. So there would be a more detailed slide of the machine learning pipeline, but basically the idea is there are a bunch of data types, right, which are fingerprints, which describe what the drug is. There are drug targets, gene ontology for these drug targets, then protein-protein interactions, and the interesting part is gene expression. So when we add this drug to a cell, how gene expression changes after we added this to a cell. So then we have many machine learning models that I will show later which is basically they can be summarized as, let's say there is some graph neural network, but for gene expression, we actually really use the MLP, even though we could use, a full graph of the cell neural network. And finally, we'll have some acquisition functions, right? Because we know this is active learning setting, we are much more interested in active learning scenarios compared to regression. So basically, I would try to focus quite a bit more on active learning. So on the data set we have chosen is actually very interesting because we have chosen something very different from what Van Gogh has chosen. So it's we basically took a very large cancer data set, and it has 500,000 cancer drug combinations. So it's really a lot of data, which is suitable for training machine learning models. It has 4,000 drugs, and the most difficult part, it has 2,000 cell lines. So cell lines are quite different from each other, so we will need to figure out a way to generalize from one cell line to another. And now are some challenges of this data set, right? So first, this data set is overrepresented, so most of these drug combinations are really on 150 drugs, right, or 300 drugs, right, which has this small basin of anti-cancer drugs. And only a few studies did drug repurposing, so which is not trying for the original target. So the data support on basically non-cancer drugs is extremely sparse. Next, if you look at so-called synergy scores, right? So when we have two drugs, we want to predict how well they work together. There are many ways to estimate what synergy score is. And there are many of them, which is S-score, ZIP score is the most common, I believe, which is basically what is nonlinear effect of drug A plus drug B. There is low and CSS and ZIP and HSA. So there are too many of them. And the thing is that we found is actually none of them correlate at all. And this is just one of the examples where we just decided to plot it. And basically synergy scores do not correlate. So if you want to run this study for real on the drugs that would be useful for the patients, we have to come up with some study or with some measure that has clinical significance because they don't correlate at all. And now the interesting part, so on this data set, I mentioned result on real cancer patient trials. So when I plotted a bunch of cell lines, and in blue here, we see the effect of a single drug combination or single drug, right? So this is the best single drug can do. And in yellow is the normalized effect of two drugs, right? So basically we say A plus B, right? And then we calculate normalized efficiency of the drug, right? So basically drug combinations in this data set are strictly doing better than a single drug. So there is, because basically whenever we have two drugs, right, we can select them to be at the same concentration. Two drugs would be more efficient than one. And you can see how pronounced these effects actually are, right? So I plotted several cell lines one by one, and we can see that the set of drug combinations is much bigger, but there is clearly much more potent inhibition of the cancer cell growth as compared to single drug on these combinations. So this is the very first model that we tried. Actually, this was tried in one night by one person. And basically what we have here is we tried the simplest model that could be. So we took one cell line at random, and then we say, okay, for the drugs, we have something called fingerprint. And fingerprint is really a hash function that is used in drug discovery. And it basically tries to break molecule into sub-building blocks and then just build a linear combination of it. So we concatenated two fingerprints. It is the easiest model we can do. And we split data into training and testing set. And we have chosen cell line for which there was 150 drugs. And this is 150 diverse drugs, and then basically 7,500 combinations. And we split them into training and testing sets, so standard 80% training, 20% testing. And these are three different cell lines, and on all of them, we actually can fit the inhibition pretty well. And what we did is next is just to know the feasibility of the study. So we fit the simplest model of uncertainty, which is basically a Bayesian ridge regression on this fingerprint, which is very straightforward. And then we try to mimic a study where we basically say, okay, if you have, let's say, a which will run 100 combinations per week. Right? So every week we would set up a new assay and run 100 combinations. Then we would basically, this would be the result. Right? And so what we see here is, I repeated this 5 times. Right? So when we see basically, and the algorithm for acquisition is UCB with kappa 0 is 0.7. So and UCB is basically mean plus kappa variance. It's a very simple algorithm. It's one of the easiest acquisition algorithms. And what we can see here is that if you do it at random, right, if you acquire random drug pairs and looking at top 1% of the best performing molecules, right, in the case of active learning, we are able to find all of them in just utilizing 1/3 of the data, right? All of the top 1% drugs are found with 1/3 of the data with a simple model. So we concluded that this would be feasible, and then we started to expand this study. So now basically we can say, okay, what are the other data types that can go in? And these data types are protein-protein interactions, right? So we know what drug interacts with, as Rafael mentioned before, and Gong mentioned in his model. Then we have drug structure, right, which is exactly the drug shape. And we know about drug targets. And finally, the most interesting part is differential gene expression, which I mentioned before. Once we add this drug to a cell, we have something called L1000 array, which can measure how much transcription has changed for each of the genes. And here is basically how relevant each of the data types by itself is. So the least relevant ones on its own is basically drug targets. And I believe the baseline model was standard 2-layer GNN. And a bit more informative is drug fingerprints. But by far the most informative on its own, a single data type is a transcription L1000 assay, which is basically when we put this drug into a cell, how transcription of 1000 genes, which are reference gene, would change. Right? So what is this particular drug doing to the cell? And we found this is really interesting because actually we can build our future studies based on this, because we do want to run active learning, and we are planning to run active learning on COVID-19 combinations. So here is a bit more complicated way on the model that I described before, right? So we basically built a bigger model, right? And it's not one model, it's like what our group is doing, like from day to day is we are trying to have basically this transcription, then protein interactions, drug protein interactions. And basically we have a joint model of all of these things together. And there are just would be too many ideas to say that which we have tried, but I will say ones which worked, right? And this is basically because we want to be practical, right? This is something that we do every 3 days or so. We're just running very large optimization of a model and just doing it at scale all the time to make sure that we do optimize hyperparameters and different data combinations as much as possible. So something we found besides, so our model for protein interactions is different from what Gong was describing, right? So we are trying to take all of the drug interactions, so all of the drug-protein interactions, and all of the protein-protein interactions. And then we have something called relational GCN, where we basically learn 2 different matrices, one for drug-protein messages, another for drug-drug messages, and another for protein-protein messages. And this in every layer, we have these 3 drug matrices. We send messages to each of the nodes, and then we compute the activation. And we do usually 3 layers of this. And usually graph neural networks, in most of the cases, actually don't go more than 3 layers. And something that we found really useful, and that's interesting. So there is something called low-run global attention approximation, or LRGA, which was designed to fix the bottleneck problem. So the bottleneck problem is we have exponential number of messages being sent to one node as we expand, as we look at bigger and bigger distances from this node. So low-run global attention approximation is similar to attention, which is very common in machine learning. So which I wouldn't describe it very well, but the idea of attention is that we have this matrix of n by n, right, of feature interaction. So low-run global attention is saying, okay, if you have n is too big to be computed, because if you have 1000 drugs and we want to compute or not 1,000, if you have 12,000 proteins or 20,000 proteins, and you want to compute the tension matrix, it would be too expensive to compute. So basically what they do is they compute principal eigenvectors of this attention. And this has been extremely useful to our study. And actually it improves performance by a very large margin. And yeah, so it's a little bit more into models of uncertainty that we use. So the best out of the results I've shown is Monte Carlo dropout, right, which is basically dropping out weights randomly and then treating the output as a Gaussian mixture model. And that was developed by, this view was developed by Yarin Gal. And here is a little bit more on the global idea. So basically, if you do active learning, right, we want to acquire data points where basically the model is most uncertain about its own predictions. And using this dropout, we can look at where the model basically disagrees most with itself, right, where the predictions are most diverse. So we'll try to acquire data points in this in the places where the same model disagrees with itself most. And acquisition functions, so here we tried greedy, which is basically choosing the best predicted value, random, and then UCB. And this is more or less one of our later results on just one cell line. And this is actually learning much better. So actually here with only 15% of the data points and GPU-UCB, we are able to recover pretty much all of the top 1% best combinations. So that's where we reached at. And one of the points that I think would be useful is also to say here that in addition to MC dropouts, the better model to use would be ensemble of MC dropouts, because dropouts are correlated to each other. So that's something we are working on right now. And I would skip this slide, but basically transitioning from somewhere where we had a machine learning model to cells was a really difficult procedure. And it took us actually as much effort as building the machine learning model by itself. I will just say a couple of words that we are trying to find a cell line in which first we have pronounced synergy or reasonable synergy, which would not be an outlier from other cell lines. And we also want to be able to predict that synergy. And finally, it should be from the data set we trust, because once we started looking into the data sets, so the data set that I was showing before is combined of multiple ones. So it appeared that some of the studies there were actually not predictable at all, and we don't know why. And finally, this is, I would say, the last slide about the future, right? So what we are doing next is actually really, really exciting. So this is, in our plan should start in January, but this is a multi-fidelity active learning, right? So we have a first degree oracle, right, which will test drug on basically COVID-19 free cell line and measure certain parameters, right, such as L1000 assay, which is very cheap. And we can do a lot of this per week. And basically, a next level of oracle, which is more expensive, is infected with COVID-19 cell line. So that cell line is by far more expensive and by far more demanding, because COVID-19 infected cell line have to go to BSL-3 lab. So they're very expensive and throughput is low. And the next layer of oracles that we have is infected microlung. So microlung, no, not infected. It's infected tissue culture. Sorry, we didn't go for microlung. So tissue culture is basically a patient tissue sample from the lung. And basically it would be taken from 10 different patients. And then we would be testing combinations on this. And finally, the most expensive oracle that we have is COVID-19 infected mouse. And we expected to have 24 infected mouse per iteration. So basically it's a very small data support on that. And basically what we would have to do is we have to design a model which can run all of the things concurrently at the same time, where we have active learning which can query one of the 4 oracles. And this is pretty difficult setup, but luckily this problem with multi-fidelity, there are some papers in literature such as Maksym research that do something similar. so solve similar problems. So there is something to start with. And finally, I probably wouldn't spend more than 1 minute on this. So thinking about what are the next steps in machine learning models that we will build. So we know that right now what we have is this graph, and this graph has, okay, let's all protein graph, then all drug graph, and then all proteins want to send messages to all drugs. And then between drugs, we have to do this basically in order to say what is the synergy between drugs, we are doing this MLP. And this is just very, very big. So in reality, what we want, and that's actually what we are building, but I don't have a result to show yet, which is basically what would work much better is a simulator, right? Because we know that this graph never changes, right, from state to state. So what we are trying to build is a cell simulator, right, which has all the proteins, all the drugs, and this cell simulator runs a transition operator in time and says how the state changes over time. And the simulator could take not only one drug or two drugs, but it can also take other data types such as knockdown experiments, which are very abundant in the literature. So this basically is the next step. And the most important point is because right now the models that we've seen before, right, which is on graph synergy learning, they don't really have something. So they try to generalize. So these graph neural networks, they were built to generalize from one graph to another, right? So if you know our setting is more signal on the graph, right? So we know that the graph would never change. And the fact, knowing that the graph would never change, we actually can do very interesting modifications, such as naming every node, right? Or for example, instead of concatenating, we can just, or instead of averaging all the nodes in the end, we can concatenate them because we know that we don't have to generalize to a new graph. And this is future work. So I believe I have time left for just one question because I went a little bit over time.
Great, thank you. Really, really impressive work. A lot of, I was really happy to learn all these exciting developments in Mila. Now, do we have any questions? And thank you for William Godinez that explained to me how the questions are sorted. Thank you, William. I learned something new. Do we have any questions for Maksym? So the audience is quiet. So I think that Maksym would be happy to answer the questions in the chat. So feel free to answer the question unless you see some. Maybe I'm not seeing something that you are seeing. Do you see something, Maksym?
I don't see my Zoom actually, but
yeah. Oh, there is a question by Noah. What's the key limitation of the cell simulator computational complexity knowledge of the biology? Try to do it in a minute.
Extremely good question. Yes, so complexity of the simulator is by far bigger than any of the graph learning model. So if you think of basically doing gradient descent, when every time we basically need to do one layer of message passing to predict all of the drug interactions, right? And that takes 1 week to fit, right? With some modifications with putting all the data on GPU, right? And basically doing it as fast as possible, it's still 4 hours to fit, right? So Decagon, Marienka was speaking before, so Decagon style of model with the best implementation we could do is 4 hours to fit. Now imagine that every time, instead of getting all of the predictions, right, like we did before, which is basically 7,000, right, we only can do one. So it's a factor of slowdown of about 7,000, but hopefully it can fit a little bit faster than that. Because basically these bottom layer features might be able to adapt faster. So we hope we don't have to do 100 epochs like regular GNNs. But it's a pretty much open question. We only had very, very early results on this yet. So hopefully I can show them sometime later, maybe after a while.
Thank you. So something for all of us to look forward to. So thank you very much, Maksym, for an excellent talk.